Welcome to the HiRSE Seminar Series on Research Software Engineering in Helmholtz and beyond.
How does the cell's fate evolve? And to answer that question, or to find an answer toward that question, we developed a tool which is called CellRank, and we now have a second version, CellRank 2, that unifies fate mapping of single cell data. So the problem and the idea that we have is that, and I'm interested in, is that we want to understand how we go from a single cell to multiple cells that then form larger structures, so tissues, for example. So one cell develops into multiple, then evolves and finds structure. These structures grow into entire organs, so for example, the heart, liver, and brain, and in the end even make up entire organisms. And what we want to understand is how this process unfolds. So this is natural development, but what we are also interested in is applying these studies then to disease. So for example, how does cancer rise and evolve? So what are the triggers? How can we prevent it? Similar like Alzheimer's disease, Parkinson's disease, and so on. And the data that we use to answer these questions, or try to answer these questions, is single cell data. Specifically, in our case, mostly single-cell RNA-seq data. What that means is that you take your data sample, so for example, a brain sample, liver sample. This gives you a collection of cells, and what you can do now experimentally is you can separate all of these cells that are in practice attached to each other and form these organs or tissues. You separate them, and now you can actually look at each individual cell and understand how it looks like in terms of specific features. So if we talk about single-cell RNA sequencing data, we look at the RNA expression, so the transcriptome in this cell. You have different options that you could look at, but I'm going to focus on the transcriptome. And what this is giving you then in the machine learning or statistical context is just a data matrix where you have the cells in for each cell. You have the expression of each gene. So is gene A turned on in the cell, and to what degree? Another way to visualize this, and how we like to visualize it, is through this Waddington landscape. And the idea is that these cells live in a high-dimensional space that looks like a landscape. So we have valleys and mountains, and higher regions are have high entropy, so less stable. Valleys are stable, so the states tend to, or you tend to find cells that live in these states. And along this landscape, we observe differentiation and development. And the task now is that, given the data we measured experimentally, to first of all align these different cell types along the landscape, and then understand, okay, how do these different cell types evolve into one another? So at the top here, for example, the dark purple cells could be stem cells. They differentiate and develop into a more mature cell type, and this cell type then all of a sudden decides to give rise to different cell populations. And of course, what we, and I hope you also ask immediately, is like these branching points. Why is this happening, right? It could be that these stem cells here, or these light blue cells here, are normal cells, and all of a sudden you something is going awry, and you find cancerous cells on the bottom here. But it could, of course, also just be normal development. So the idea that we have is, or the standard process we have, I thought I just walk you through this once, is you have your original data. The first thing you do is, as in any machine learning or data processing pipeline, is do transportation. So you normalize your data and so on. You select features. In our case, it's genes that vary across the entire sample, because we're only interested in genes that vary. If a gene is doing the same thing in no matter which cell it is, it's not going to tell us anything about what's different between these cells. After we selected the most informative genes, we reduce the dimensionality, because in the human body you have about 30,000 genes, so that's way too many features, and it's very sparse. And once you've reduced your dimensionality to something more tractable, you now start comparing these different cells. So for each cell, you look what cells are similar to it, and thereby you can, for example, construct a neighbor graph. So this is where we come in and want to aid in inferring the cellular fate. What we want to do is you go from a physical sample, you construct a data representation, as I showed, for example, the neighbor graph, and now you want to understand, given a cell, so the center cell, for example, this reference cell, what is its likely future state? What I didn't tell you is that for each cell, you could just argue, okay, I'm just going to look at this cell and see how it changes over time. The caveat is here that you can't do that, since once you've measured the cell, it's destroyed. So what you have to do next is then, once you've quantified how likely other cells are the future state of your representative cell, you want to infer the fate and thereby say, okay, cell type purple is transitioning into blue, into red. We aren't the first to ask this question, so there are other approaches where you assign cells along the developmental trajectory and thereby infer directionality. Another approach is based on mechanistic modeling, where you try to model the splicing kinetics. So DNA is transcribed into RNA, which is then transformed a bit further. And with experimental improvements, you can nowadays also have more complex data sets which have temporal information, and there you can use concepts of optimal transport. Sorry. The problem of these methods was always that they are limited to a single view, and thereby you can't really generalize. And whenever experimentalists or someone else comes up with a new view, a new data type, you always had to go back to the drawing board and construct a new method to infer cellular fate. So what we did is we realized that given all these different concepts of quantifying how cells evolve, you just need one of these. As a next step, you design a cell-cell transition matrix. So that was the step that was saying that for each reference cell, you find likely future states. And following that, you can infer future states. This is done with our estimator. So at the moment, we only have one, but you could choose a different one. The idea here is that you use Markov chain theory by going from a high-dimensional space into a lower-dimensional space, solving the problem in the more tractable lower dimensional space and going back to the high dimensional space. So once you've done this, you can actually automatically infer terminal and initial states in your data. You can assign fate probabilities towards, in this case, terminal A or terminal B state, and you can study how these gene expressions change along this trajectory. I don't want to dive into too many details here, but the main message is that we have one method that scales much better than comparable methods. In our case, we can easily run this on a data set which has over 1.3 million cells. And we'll use this method to study embryo development. Specifically, we were interested in how the endoderm arises, and what we can do with our software is now see how gene expression changes as cells develop into an endoderm cell. And you already see that these genes turn on and off, and thereby we could find several genes that are relevant for this emergence of the cell type. Similarly, for time-resolved data, we have a new method that allows us to granularly map fates by using both the information crossing time points as well as the information that we already have in a single time point. And to showcase this, we looked at pharyngeal endoderm development. The pharyngeal endoderm is crucial for organ development, for example, for the thymus. Again, we had several time points, and we focused here on the emergence of a cell type called medullary thymic endothelial cells. And we focus on this because little is known how these different cell types actually arise. And once we used our tool to study the data, we found that in this cluster of cells that was only annotated as progenitors, we found a state with high probability of turning into MTEC cells. And when we checked why these cells are turning mTOR cells, so which genes are most likely to cause this, we found a lot of genes that have been known and mentioned in literature, as well as transcription factors, which are driving these processes. Similarly, if you compare it to a previous approach that only considered the information across time points, you couldn't achieve the same performance and gain the same insight. Now, I just want to briefly show that our concept also generalizes to new views. So, for example, we applied our data, our concept to study Alzheimer's disease, and we could show that a specific cell type is more prevalent and relevant for later stages of Alzheimer's disease. And we could also show that as disease progresses, the biggest jump in disease severity seems to appear between these stages 3 and 4, which was also confirmed by pathologists. Similarly, you now have other papers and other works using our tool in their pipeline. So this shows that it's modular and usable by the community. For example, in this case, they didn't only use gene expression, but also lineage information that you can measure experimentally. And with that, I would just like to thank my co-authors and co-developers, especially Maurus and Mike, who were detrimental for this. The PIs, Fabian, Thijs, and Dana Per, who were involved in the study. On the right here, I've linked the QR codes to the paper and the software documentation. I would like to thank everyone that helped us during the development of this project, and for the Alzheimer's disease data, of course, would especially thank the donors and their families for their contributions, and finally also the lab that I'm affiliated with. And with that, I'm happy to take any questions if there are already any.
Thank you, Philip. Thank you very much. We will actually move on to the next speaker, that's okay, and take the questions after our next two talks, if that's okay. But thank you very much for that. Okay, our next speaker is Birgit Hassler from DLR. They are the winner of the Sustainability Prize. Their system is called HiRSE System Model Evaluation. And Birgit, if you could please share your slide.
Mm-hmm. Well, thank you so much for having me and for letting me talk about the HiRSE System Model Evaluation tool, in short, ESMVal tool, that is a community-developed tool to analyze climate models. So the motivation behind our software development was that we have HiRSE system models, in short, ESM, that are important tools for improving our understanding of the climate system and projecting future climate change. But to understand how those HiRSE system models, ESMs, actually work, we have to evaluate them. We do that with Earth observations, like satellites or airborne data. And that evaluation is crucial for making sure that we improve the models constantly, that we have a better process understanding of the climate system, and that we have more trustworthy climate projections that can be used for policy guidance. And at DLR, we are using the HiRSE system models for technology assessments in aeronautics, transport, and energy. The challenge there is, however, that climate models increase in complexity and resolution over their different generations over the years. And that makes it harder and harder to get all the information that you want out of them and intercompare them. So this is where the ESMVal tool comes in. ESMVal tool is a community diagnostic and performance metrics tool for the evaluation and analysis of HiRSE system models. And at the beginning, it was primarily focused on a multi-model context. And you will see in a bit what I mean with this. It's development, it's community-driven, it's open source, and it's on GitHub. We have more than 200 developers, more than 60 international institutes contributing to the code and to the tool. And its development is funded by many national and international projects. We are very proud that ESMVal tool was used in several chapters of the assessment report 6 of the IPCC's Working Group 1, to produce some figures for that very important report. And in 2020, we released version 2, and I will get into that a little more in detail as well. The release of version 2.0, and we are currently at the version 2.11. So you can see on the bottom here a timeline on our regular releases that we have. We have maybe 2 to 3 a year, and we try to keep track of improvements, updates, and new developments from the community. So on the right, you can see 5 papers that describe the scientific documentation of ESM-Val tool with different foci. So the first one is probably the most interesting one for research software engineers. It's the technical overview on how this tool actually works. But before we go to the technical details with a flow chart, I wanted to tell you a little bit about the historical development of ESM-Val tool. We had a version 1 that was officially released in December 2015. It's based only on open source languages with code being publicly available. And the idea was that the analysis code for multi-model comparisons, different analysis, where you look into different models and try to find the commonalities, that code is available for everybody, and it does not have to be reinvented over and over, and gives therefore the scientists more time to actually think about different things than about how can I rewrite a code that had been already used 4 years ago. So in version 2, from 2015, the tool was continuously developed, and we realized we have to radically overhaul it to make it adjusted to the new approaching challenges. And so the overhaul was finished in 2020. That was when we released version 2 of ESM-Val tool. And at that point, we split ESM-Val tool, the whole package, in an ESM-Val core, which is Python only, and ESM-Val tool, which still supports different open source languages. We moved the code to GitHub and introduced software engineering guidelines, and you can see some of them on the right side here. For example, automatic code checks, automatic testing, and then we updated our documentation and introduced an online tutorial for new users to make sure that people actually can use it easily. It still has a learning curve, but with the online tutorials, it actually helped quite a bit to make it more accessible for new users. So this is the workflow basically in a few boxes. We have the input data on top. This is the blue box. That could be model data, could be observations, could be models in native formats. That data is read and introduced to ESM-Val core, where there will be all kinds of different pre-processing steps available that the input data could go through. For example, we could have like different regridding schemes. We could mask land, sea, ice. We could have time or space extractions. We can mask missing values. We can vertically interpolate. This is all these pre-processing functions that are the main part of ESM-Val core are very important when you think about a multi-model intercomparison. For that, those model simulations that are not necessarily on the same grid, on the same levels, need to go to that same grid and the same levels to be intercomparable. And with these preprocessor functions, and we can make sure that all the data goes through the same steps, and nobody has to kind of write the code for missing value masking again and again. So if we have the preprocessed data ready, it goes into the next dark blue box. And then from there in the red box, which is the heart of ESM-Val tool, is the diagnostics packages. Can be, as I mentioned, different open source languages. And there the science happens, what we want to see from the data. And then from the diagnostics, we go to the right dark blue box where says output. That can be graphic files, netcdf files, log files. You basically, as a user, define what you want to get out in the diagnostic. driven is ESMVal tool by a recipe. That's the purple box, and kind of like it, you can tell it exactly what to do and where to look for data and stuff with different configuration files. So we have information available for ESMVal tool. As I mentioned, we develop it on GitHub. So we have different GitHub repositories where you can find everything. All code that makes ESMVal tool and ESMVal core is available on GitHub. So you can see everything there. We have a documentation that's on Read Docs, and it's very massive. You can find any information that you could possibly want about how to run ESMVal tool, how to configure it, how to install it, how to kind of like what kind of recipe to use, what data sets are available. It's all there. It's sometimes a little hard to find, I heard, but we are constantly trying to improve that to make sure that it's easy for people to use and to understand. We have that online tutorial that I had already mentioned, and we have a web page where you can find the kind of like general overview topics about that ESMVal tool, for example, is at EGU, and you can, we have a breakout session there, and you can come and talk to us. We also have a Twitter channel, which is, or X channel, which is sometimes more active than other times, but that all depends on what's going on in the community. We have, since ESMVal tool is basically a science-driven software, so it's on a crossroad between technical development and science development. We have, of course, science contributions, and I wanted to mention a few here. We have more than 60 scientific peer-reviewed publications where ESMVal tool has been used for the analysis. As I had mentioned, we have a lot of projects, national and international science projects that fund the development of ESMVal tool, including large EU projects. And we are especially proud that ESMVal tool has been used for the production of figures for several chapters of the assessment report 6 of the IPCC's Working Group 1, and also including provenance record, which is a very important part for IPCC now. You can see here the front of this IPCC report, and on the left you can see one of the figures that has been produced with ESMVal tool. All the tiny wiggly lines are individual model simulations of near surface air temperature, and then all this data have been read into ESMVal tool, have been, the anomalies have been calculated. Then you plot all the different lines here, and we have the multi-model mean, which is the red line, and the black line is an observational data set. And you can see kind of how the anomalies of near surface air temperature develops over time. DLR contributes to ESMVal tool with one PI, that's me. We have two PIs. The other PI sits at the Met Office in the UK. We have several core developers, and we evaluate DLR developed models and the national model ICANN XPP, for example. So we use also ESMVal tool to compare different classes of climate models. And so one of the developments that we're doing here is that we develop machine learning based climate models that help reduce systematic errors and long-standing biases. For example, what you see on the left side is a biases of a diurnal cycle of precipitation that has been improved with one of those machine learning based climate models. And we also improved the extreme precipitation representation in one of those model simulations. So what's next for us? Further, of course, ESMVal tool is constantly further developed with that many people actually working on it and using it for all different kinds of things. I just listed a few things here that we are actively working on right now, and it's not like in a distant future or close future. We are working on extending the monitoring and benchmarking capabilities of ESMVal tool so that it can it can be more helpful and useful for model development, which is, of course, a big part. You want to see when you develop a model how your simulation holds up, and if you have to restart things. And the earlier you can actually notice that, the better and the more sustainable that development becomes. We are constantly improving or upgrading our satellite observation representation or the pool of observations that we have available. And one of the implementation of ESA EarthCare and DLR CNS mission Merlin is in our near future as soon as the data is available. We expand ESM tool with new pre-processing capabilities and also, of course, new diagnostics, because that's what the scientists work on. And that's what the scientists use the tool for. And then, of course, we are trying also to make sure that we are ready for using the next generation supercomputers and data infrastructures. So we are trying to reduce the memory footprint to read more and more memory heavy data into ESM tool to be able to work with a lot of different data sets and also high resolution data sets. We are introducing parallel tasking for the same reason. And then ESM tool is central for the upcoming intercomparison projects. For example, the coupled model intercomparison project, which is called CMIP. CMIP has decided that they want to have a rapid evaluation framework, in short REF, that they want to have for the next phase of inter-model comparison, and ESM tool will play a central will be one of the central components for that, and that is all basically in preparations for the next IPCC report. And with that, I want to thank you for your attention, and I'm happy to answer any questions.
Hopefully you can hear the applause from here. I don't know if it comes across to Zoom.
It doesn't, but I can see people clap, so that's good.
Okay, that's great. Thank you very much. We'll take some questions at the end, if that's okay. So please don't leave us just yet. So our third and final speaker is Marie Houillon from KIT. They are the winner of the Newcomer Prize, and their software is called OpenCarp.
Thank you. Hello. Thank you for being here to hear about the OpenCarp software, which is indeed the winner of the Newcomer Prize at the Helmholtz Software Awards. So I am Marie Houillon, and I work as a research software engineer at the Karlsruhe Institute of Technology. And I am one of the maintainers of OpenCarp. The other current maintainers coming from various places, such as Graz in Austria, Bordeaux in France, or Valencia in Spain. Unfortunately, there are too many of them so that they displayed on this slide, but it's all available on the OpenCarp website. So first of all, what is OpenCarp? OpenCarp, it is an open cardiac electrophysiology simulator for in silico experiments, and it offers to perform electrophysiological simulations of the cardiac tissue from the scale of the ion channel and single cardiac cells to the whole heart level. And additionally, OpenCarp also includes a wide variety of functions for pre- and post-processing of data, as well as visualization. I won't go into the details of the mathematical formulation or solvers implementation today, but for example, at the tissue level, the simulator has three main components. The first being the parabolic solver, which will determine the propagation of the electrical activity from the extracellular electric field and the current state of the transmembrane voltage. There's also an ionic current component, which describes the ionic transmembrane currents. And the third one is an elliptic solver, which will compute the extracellular potential from the transmembrane voltage at each time instant. For the rest of the presentation, I will more focus on the software aspects of OpenCarp. But before that, a few use cases for OpenCarp. So it can be used, for example, for helping to generate digital twins for personalized medicine and in silico clinical trials. It can help to generate large data sets that enable machine learning studies, also for benchmarking of tools for diagnosis and therapy, and to have better mechanistic understanding of disease mechanisms. And here you can see some illustrations taken from such studies. So after this general presentation of OpenCAP, I would now like to go more in details into the different components of the software. So OpenCAP is actually a whole ecosystem, and its core is the OpenCAP simulator, which is a C++ program that is free for academic, non-commercial use. And this simulator is what allows to perform simulations from the single cell level to the whole organ level. In addition to this core simulator, there is a Python framework named Caputils that has been developed, and this Python framework facilitates the interaction with the C++ simulator, and it also allows to develop complex simulation pipelines from the mesh generation and pre-processing steps to post-processing and visualization, including, of course, all modeling and simulation steps. In addition to these two main components, the OpenCAP ecosystem also comprises a set of examples, as well as a large community platform composed of the OpenCAP website, the OpenCAP GitLab, as well as a question and answer system where users can get help from the OpenCAP community. So now let's focus a bit more on technical aspects of OpenCAP. From a user's point of view in practice, the simulator is composed of several command line programs. There are two executables that are the actual simulators. Bench allows to achieve single cell simulations, and the OpenCAP executable allows to achieve tissue level simulations. And there are also some pre- and post-processing tools, Mesher for generating meshes, and these IGB executables that allow to manipulate the output files produced by OpenCAP in IGB formats. I don't have time to expand on the whole software architecture here, but here is a picture to get a whole picture of it. What we can see in particular is that OpenCAP interoperates with various standard formats like CellML to import cellular models. It also interoperates with MyoKit, a Python software package that is used for the analysis of cardiac myocytes. And OpenCAP also interoperates with several file formats, as shown on the right here. And for this, it uses two other software pieces that are also part of the OpenCAP ecosystem. Mesh tool for mesh manipulation, and Meshalyzer for visualizing time-dependent data on 3D finite element meshes. Aside from the software itself, there are also many examples that are available and can be used as tutorials for new users. In particular, there are many experiments coded up with Caputils at single cell level as well as at tissue level. And several examples also include pre- and post-processing steps. Another important part of the OpenCAP is its community platform, which includes a regular newsletter, in-person and online user and contributor meetings, a question and answer forum, as well as a software issue tracker on the OpenCAP GitLab. The user community of OpenCAP is also built on contributions to the software from users and users that share the experiments that they've created using OpenCAP within the community. The last but not least part of OpenCARP that I would like to develop is the infrastructure that is in place to ensure the maintainability and sustainability of the software. OpenCARP is built on a collaborative development environment based on GitLab, which includes an extended DevOps pipeline for continuous integration and delivery of the software. So in terms of maintainability, OpenCARP relies on version control using Git, and it uses code review for new contributions to the software. And one of our main concerns for improving its maintainability is to reduce the load of maintenance tasks that have to be performed manually. And in order to achieve this, we have automated regression testing that is achieved using the GitLab's continuous integration and delivery pipelines. And our pipelines also perform automated benchmarking, automated package generation on various platforms, as well as automated generation of documentation and website content. And in addition to this, the sustainability of the software is also enforced by ensuring long-term preservation of the software by publishing all releases on a persistent research data repository, namely the RADAR service, according to the FAIR principles. That means in particular that all software versions are citable, described with relevant metadata, and associated with a persistent DOI. And this publishing process, it's actually quite straightforward in OpenCARP, as it is also mostly automated using the continuous integration and delivery pipelines. So this marks the end of my presentation. If you want to know more about OpenCARP, I invite you to go to opencarp.org or to reach us by email at info@opencarp.org. I thank you very much for your attention, and we would also like to thank the Helmholtz Association for valuing research software through these software awards. Thank you.
Thank you for watching our seminar. Please check out the slides on Zenodo linked below and see you next time.